When a baby drinks milk, the mother's body reads the baby's saliva at the point of contact. Within hours, the milk changes, producing targeted defenses for whatever the baby encountered that day. The mother cannot feel this. The baby cannot know it, but the milk does. The way this works is not magic, though it is easy to mistake it for something like that. At the surface where feeding happens, a tiny amount of the baby's saliva makes contact with the mother's body. That saliva carries traces, invisible, chemical traces of whatever the baby's mouth has been near that day. Bacteria, viruses, dust, the particular air of the room. The mother's immune system reads those traces. It already knows most of them. The unfamiliar ones it responds to. Within 6 to 8 hours, the milk arriving at the next feed contains a higher concentration of the specific protective proteins aimed at whatever the baby was exposed to. The milk has been updated, not by thought, not by intention, by the quiet chemistry of two bodies in contact, exchanging information in a language that requires no words and leaves no visible record. Think of it as a letter written without ink. The contents of the letter change depending on who will receive it and what they were doing the day it was written. The envelope looks the same every time. The inside is different. What travels between the two bodies is not visible at any surface, audible in any sound, or detectable by any ordinary sense, and yet it is specific, it is timed, and it is aimed precisely at the circumstances of the individual receiving it. No two feeds are identical. In the same way, no 2 days are identical. The letter is rewritten every time. This happens in ordinary feeding every time, without ceremony. It is the kind of care that is invisible to the person receiving it, because the person receiving it has not yet learned to look. The glass on the table holds no memory of this. The milk in it cannot respond anymore. But the structure that produced it was capable of that precision, and the architecture of that precision is still present in the liquid, readable at the right scale. What the eye sees first, before any of that, is the color. Milk is white, not off-white or pale or cream. A full, complete white, one of the more reliable whites found anywhere in the natural world. It does not come from any dye. It is not bleached. There is no white ingredient added to it. The whiteness is entirely a consequence of what happens to light when it enters the liquid. Think of standing in fog. The fog has no color of its own. It is simply water droplets suspended in air, yet it appears white. Light enters from every direction and bounces around inside the fog, changing direction at each droplet, fanning out rather than passing through. By the time that light reaches the eye, it has scattered so many times that all the colors it carries are mixed equally. The result is white. The fog did not add anything. It simply redirected what was already there. Inside a glass of whole milk, there are approximately 10 billion tiny spheres of fat. They measure between a fraction of a micrometer and 10 micrometers across, the smallest among them so fine that 1,000 placed end to end would barely span the width of a single human hair. Each sphere is wrapped in a thin, organized skin, the way a soap bubble has a surface that separates inside from outside. When light hits one of those spheres, it does not pass through, and it does not reflect cleanly. It scatters, fans outward in all directions, the way a beam of light spreads when it hits a frosted window. That scattered light then hits another sphere, and scatters again. With 10 billion spheres, every color of light is redirected equally. No wavelength is preferred, none is absorbed. The result, seen from the outside, is white. This is identical to what produces the color of clouds. A cloud is water droplets floating in air, each droplet roughly the same size as the fat spheres in milk. They scatter every wavelength of light equally. Clouds that we see almost every day, drifting above our heads, watching us, moving here and there without ever saying they are tired, and the cold white milk we take from the refrigerator in the morning and drink with such satisfaction are the same optical event. They may be different materials from one another, and regardless of the environment they exist in, they operate through a single mechanism. Two very different things arriving at the same color through identical physics is worth staying with for a moment. It means the white of milk is not a property that belongs to milk specifically. It belongs to geometry, to the relationship between a particle's size and the wavelength of the light that meets it. Change the size of the particle, and the color changes with it, regardless of what material the particle is made from. That white is not identical in every form of milk. If we remove the fat from milk, separating them from one another, it takes on a faint blue tint under sunlight. We can only observe this in a regular clear glass and in an environment with good light. When the fat spheres are gone, the particles remaining in the liquid are primarily the protein clusters that give milk its structure. These clusters are smaller than the fat spheres, much smaller, and smaller particles do not scatter all colors equally. They favor blue. This is because blue light has a shorter wavelength than red or yellow, and shorter wavelengths interact more strongly with smaller particles. The protein clusters scatter the blue more strongly than the rest. The result is a blue tint at the surface of skim milk. The sky is blue for the same reason. The molecules in the atmosphere are far smaller than any droplet in a cloud, and they scatter blue light so thoroughly that it fills the entire sky. Skim milk in a clear glass placed near a window and the clear afternoon sky above it are optically the same type of event, scattering from very small particles, favoring the short end of the visible spectrum. Neither was designed to be that color. Both are displaying the physics of particle size and light wavelength, operating at very different scales but governed by identical mathematics. In whole milk, this blue tint from the protein clusters is there but invisible, overwhelmed completely by the total white from the fat spheres, which scatter so much light from all wavelengths that the blue signal cannot be detected. Remove the fat, and the blue emerges from underneath. What looks like one uniform color in a glass of whole milk is in fact two competing light effects coexisting, the fat dominant, the protein hidden below it, waiting quietly to show itself if the fat is taken away. The color is one surface reading, weight is another. Milk feels heavier than water in the hand. The glass takes slightly more effort to lift than an equal volume of water would. The difference is small but present, The dissolved proteins, sugars, and minerals add density to the water that carries them. This is also audible. Milk poured from a height into a glass sounds different from water poured the same way. The sound is lower, softer, more continuous. The liquid is reluctant to break into droplets. Pour cold, whole milk from a glass jug, and the sound of it is heavier, a thicker, more unbroken stream than water, a foam at the surface that holds longer before it flattens. At the moment of contact with the glass, the surface disturbs differently, less splash, more spreading, the way a heavier material finds its level with less violence. The sound of milk pouring into a cold glass in a quiet kitchen is one of the more distinctive sounds in an ordinary household. Lower than water, continuous, almost silky in the way it connects pore to surface without interruption. That foam is worth a moment. Whole milk shaken or poured vigorously produces bubbles that are smaller and more stable than water bubbles. The proteins dissolved in the liquid immediately gather at the surface of each bubble as it forms. They build a thin film around the air pocket, a wall that slows the bubble's collapse. A water bubble has no wall. It pops in under a second. A milk bubble has a protein wall that holds for 30 seconds to a minute before it thins enough to break. This is exactly why steamed milk in a coffee shop holds its foam. The heat and pressure help the proteins already in the milk build more and better walls more quickly. The barista is not adding anything. The material is already capable of it. The foam it produces is simply the surface expression of what was always present in the liquid, given the right conditions to appear. Beneath that foam, the fat spheres are in motion. At room temperature, the water molecules surrounding every fat sphere are moving fast, faster than a car on a highway. Each one collides with the fat sphere billions of times per second. Most of those collisions cancel each other out, but at any given instant, there are slightly more hits from one side than another, and that imbalance pushes the sphere on a slow, continuous, unpredictable walk through the liquid. The path cannot be predicted ahead of time. It can only be described in terms of probability, where the sphere is likely to end up, not where it will definitely go. The physicist Albert Einstein worked out the mathematics of this motion in 1905, using the same equations that apply to pollen grains drifting in a pond. The prediction matched the measurements exactly. The fat sphere wandering in a glass of milk follows the same rules as a grain of pollen in a still pond in summer, both driven by the same thermal energy, both following paths that can be described statistically, but never traced in advance. The motion is not random in the sense of being arbitrary. It is random in the sense of being genuinely unpredictable, a distinction that takes a moment to sit with. A glass that appears perfectly still contains 10 billion such walkers, all moving, none visible. The stillness is a surface reading. There is one more visible quality of the liquid that is easy to overlook. Whole milk is slightly viscous, slightly reluctant to flow. This is not dramatic. It does not pour like honey or syrup, but it does not move like water. It has a mild resistance, a faint thickness that comes from the protein clusters and fat spheres occupying the liquid and adding internal friction. This viscosity drops as the milk warms. If we take milk at room temperature and milk freshly taken from the refrigerator and compare them, Even if we can barely feel it, we can see that the milk from the refrigerator is slightly thicker. The difference is so small that it can sometimes escape our notice. That is why most people do not feel it or pay attention to it. I say this as someone who has one day observed this with my own eyes. Warm milk pours slightly faster. Cold milk falls more slowly and coats the inside of a glass with a faint, even layer that water would not leave. A pale, thin film that holds for a few seconds before it runs down and disappears. It is the kind of detail noticed only when one glass is compared directly against another, cold against warm, side by side. The cold glass against the palm carries that slight extra weight, that barely there chill that lingers a half-second longer than water would. The density announcing itself quietly before anything else is known about what is inside. At the molecular scale, that resistance is the physical sum of every protein cluster and fat sphere pushing through the surrounding water with each motion of the glass. The liquid is not uniform inside. It is a moving population of objects suspended in the water that carries them, each one contributing fractionally to the drag on the pour, the faint coat left on the inside of the glass, the sound that is lower and softer than water when it finally meets a surface. What the glass holds, then, is a liquid that is white because of geometry, heavy because of dissolved solids, slightly resistant to flow because of what is suspended in it, and producing foam because its proteins build walls. All of these properties visible at the surface, none of them requiring any knowledge of what is beneath the surface to observe. The surface, as it turns out, is a very thin description of what is actually in there. Before milk reaches a refrigerator, it passes through two industrial processes that most people would not think twice about, and that each leave a permanent physical mark on the liquid, even though neither one changes what the milk looks like from the outside. The first is called homogenization. Without it, a bottle of milk left undisturbed for several hours would begin to separate. The fat spheres, slightly less dense than the watery liquid surrounding them, would slowly drift upward, accumulating at the top of the container in a thicker, richer layer. This is cream. The separation is simply gravity doing what gravity does to two liquids that are not equally dense. Before the 20th century, this was expected. A line forming at the top of a bottle of milk was not a defect. It was physics working correctly, in plain sight, every morning. Homogenization stops this from happening. The milk is forced at very high pressure through a gap that is smaller across than a single human hair. The fat spheres do not survive the passage intact. They are disrupted by the force, torn apart into fragments far smaller than the original. What was a sphere measuring up to 10 micrometers exits as many fragments averaging around 1/10 of a micrometer. The physics that governs the rate of upward drift scales with the square of the sphere's radius. At 1/10 the original size, the drift rate falls to 1/10,000th of its original speed. The visible cream line disappears not because anything has reversed, but because the law that produces it is working against a geometry too small for the effect to register within a human time frame. The physics of separation still operates. It has not been switched off. It has been made so slow that it no longer matters within the time frame in which anyone is likely to drink the milk. The result is a glass of milk that looks uniformly white from top to bottom every time, without variation, a consistency that required an industrial solution to achieve what the biology had never prioritized. What happens to the surface of each fat sphere during this process is a separate story. The original coating around each fat sphere, the thin, organized skin that separates the fat from the surrounding water, is not designed to cover fragments 100 times smaller than the original. There is simply not enough of it. The newly formed smaller spheres are immediately recoated by whatever surface-active proteins are floating nearby in the liquid, primarily the casein proteins, which are present in abundance and which are attracted to fat-water boundaries. The new coating has more protein and less of the original lipid material than the native version. The fat content of the milk is unchanged. The caloric value is unchanged. The whiteness is unchanged. What is different is the surface chemistry of each fat sphere, a change that cannot be seen, tasted, or felt, but that is detectable under chemical analysis. The milk in a carton from a supermarket shelf is not wearing its original coat. Every fat sphere inside it is wearing a substitute, a secondary arrangement assembled from available materials at the moment of disruption, stable enough to last, different enough to matter under close examination. The reason cream appears yellow is a different story, and it begins in the field. The fat in milk is not colorless when it concentrates. Cream is noticeably yellower than whole milk, and the color comes from something the cow ate. Green plants contain a pigment called beta-carotene, the same compound that gives carrots their orange. The cow absorbs it from grass and forage, and it moves into the fat fraction of the milk. In whole milk, the concentration of this pigment is so diluted by the fat sphere's scattering of all light wavelengths that the color is invisible. The white overwhelms it. When fat concentrates into cream, the pigment concentrates with it, and the yellow becomes visible. A cow that eats fresh summer grass produces richer and more vividly yellow cream than one fed on winter feed. The color of the cream tells of the condition of the pasture, of how the animal was fed during those days. When cream is churned into butter, that yellow concentrates further. A stick of deeply yellow butter in July, and a pale, almost white stick of butter from a cow on a winter diet are not different products by any processing standard. Despite being from different times of the year, they are the same product, but the color tells of what the animal ate. Butter can appear in different shades depending on the season, and through these colors we can know which season it belongs to. Heat introduces a different and equally permanent kind of change. Pasteurization heats milk to 72 degrees Celsius for 15 seconds, then cools it rapidly. This is the standard commercial treatment, and it reduces the bacterial population in the milk by approximately 99.9% without eliminating it entirely. The casein protein clusters survive this temperature with their structure essentially intact. They are robust enough, held together by enough calcium bridges and electrostatic forces to come through the heat treatment unchanged. Their behavior during cheese making and fermentation is not significantly altered by standard pasteurization. Whey proteins, The proteins dissolved in the liquid phase of the milk, which make up approximately 20% of milk's total protein content, are a different story. The most abundant of them is shaped at room temperature like a tightly folded package. Think of a protein molecule the way you would think of a long thread of paper folded into a very specific three-dimensional shape, everything tucked inward, every crease in an exact position, the whole thing small and compact. At 72 degrees, the folds begin to release. The thread starts to open. This unfolding is called denaturation, and it is not reversible. The protein will not refold into its original shape when the milk cools. It is like creasing a sheet of paper. The crease can be pressed back, but the minery of it remains. Once opened, neighboring unfolded proteins can link to each other through bonds that did not exist while they were folded. Some of these linked proteins attach to the surface of the casein clusters, changing their surface chemistry. This is why heated milk behaves differently from raw milk during fermentation. The changed surface of the casein cluster means it interacts differently with the acids produced by bacteria. The gel that forms in yogurt made from pasteurized milk has a finer, more tender structure than yogurt made from raw milk. The difference in texture between a gently set yogurt and a coarser, more broken one often traces back to this protein unfolding during heat treatment, an event that is invisible in the final liquid, but present in every structural consequence thereafter. The heat left a signature in the protein, and the protein carries that signature forward into everything made from it. Ultra-high temperature processing goes further, 135 to 140 degrees Celsius for 2 to 4 seconds. This sterilizes the milk entirely, extending shelf life to several months without refrigeration. At those temperatures, minor reactions occur in the sugar fraction of the milk that produce a faint, cooked taste, detectable to anyone who has noticed the particular flavor that comes out of a long-life carton. These trace compounds are present at concentrations too small to affect nutrition in any measured way, but they can be identified chemically, and they can be used analytically to distinguish ultra-high temperature milk from standard pasteurized milk. The slight taste difference between a carton from the refrigerated section and one from the shelf is measurable, and it has a chemical address. It is a taste that belongs entirely to the heat, not to the animal, not to the grass, not to the season, just to the temperature the liquid reached and what that temperature quietly rearranged. Inside a sealed container, a slower change is also already underway, one that begins before any visible sign appears. A small residual bacterial population survives standard pasteurization. Those bacteria are not dormant. They are metabolizing, slowly because the refrigerator suppresses their activity, but continuously. They consume the sugar in the milk and produce lactic acid as a byproduct. The acid accumulates gradually. The milk has a natural buffering capacity. The proteins and minerals resist the pH change, absorbing the acid and keeping the liquid stable. But this buffering is finite. The milk can hold the acid at bay for several days. Then, the pH falls past the point where the protein cluster's surface charges are neutralized and the clusters begin to aggregate. What looks like sudden curdling, the seemingly rapid change from liquid to clumped, has actually been building for days beneath the surface, invisible, consistent, and chemically inevitable. The white surface gave no indication. It looked the same on the fourth day as it did on the first. The smell arrives before the visual change. Short-chain fatty acids released by enzyme activity in aging milk, the same acids responsible for the sharp smell of rancid butter, become detectable before the texture shifts. The nose has a lower threshold for these compounds than the eye has for cloudiness or separation. By the time the milk looks wrong, it has smelled wrong for some time. These are the same short-chain compounds that, at much lower concentrations in fresh, warm butter, produce the characteristic richness of the flavor. The smell of butter is pleasantly appealing to us, right? But what about the smell of old milk? The smell of old milk is not pleasant to us at all, is it? It is fair to say that while most people find the smell of butter nice, the smell of old milk is pleasant to no one. Yet the pleasant smell of butter and the smell of old milk come from the same chemical substance. If you were to ask how this can be, we can say that the molecule is not two different things. It is the same thing in two different amounts, meaning when present in small amounts, it is not a problem. It becomes appetite opening. But when present in large amounts, it becomes unsettling. What the refrigerator is doing precisely is slowing all of this, not stopping it, slowing it. The bacteria that survive pasteurization have optimal temperatures between 25 and 37 degrees Celsius, the range of a warm room or a human body. At 4 degrees, their metabolism is reduced to a small fraction of that rate. What would take 12 to 18 hours at room temperature takes days in the cold. The labeled date on a carton of milk is not the date at which the chemistry stops. It is the date beyond which the product falls below the palatability standard under expected storage conditions. The chemistry continues past that date. It was never not continuing. There is a mammal alive today that produces milk without nipples. The platypus feeds its young by seeping milk through pores in the skin of its abdomen, the way sweat moves through skin, except that what emerges is milk. The young press their bills against the mother's fur and lap from the surface. There is no nipple. There is no structure to latch onto. There is a porous surface and a liquid appearing through it, pooling in the grooves between patches of fur, waiting to be found by a creature the size of a thumbnail that navigates entirely by touch and smell. The feeding is unhurried. The geometry is ancient. And if the image of it seems strange, that strangeness is largely a matter of what we consider normal, because what most mammals do is simply a later variation on the same basic idea, elaborated by time into a more familiar form. This is not a broken or primitive version of what mammals do. It is a different solution to the same problem, one that has been working for tens of millions of years without modification. The platypus belongs to a group of mammals called monotremes, which includes only 5 living species. They are the only mammals that lay eggs. They diverged from the rest of the mammalian lineage before the group that includes humans, cows, dogs, and elephants had appeared in any recognizable form. The platypus retained egg-laying. It also retained skin-seeping milk delivery. The two strategies work together. The egg provides the initial nutrition in a protected shell, and the milk through the skin provides nutrition after hatching, without any intermediate structural evolution toward a protruding nipple. What this reveals, when taken alongside other evidence, is where mammary glands came from in the first place. The leading hypothesis, supported by genetic evidence, though not without debate among researchers, is that mammary glands are modified sweat glands, specifically a type of sweat gland called apocrine glands, which produce a richer, more protein-dense secretion than ordinary sweat and are still present in human skin, primarily under the arms and in a few other locations. The genetic instructions used to build a mammary gland and the genetic instructions used to build an apocrine gland overlap substantially. Several genes that are active in mammary tissue during lactation are also active in apocrine gland tissue. The developmental pathways share common elements. Two structures that look and behave nothing alike are built at the molecular level from related sets of instructions, like two buildings with completely different purposes that share most of their architectural blueprints. The blueprints were not written twice. One was adapted from the other, slowly, across enormous stretches of time. If this is correct, then the transition from sweat gland to mammary gland was not a single event. It was a long series of small changes over millions of years, each one slightly more useful to the young that received it, each one preserved by the advantage it conferred. There was no moment at which a sweat gland became a mammary gland. There were incremental modifications that eventually accumulated into a liquid specific enough and rich enough in nutrients to be called milk. The platypus in this story represents an early configuration in that sequence, a body that has advanced past plain sweat but not yet arrived at a nipple. It is not behind, it is on a different branch of the same tree, one that had no reason to change because the strategy it was already using worked. The diversity of what milk looks like across mammal species makes the word milk feel like an oversimplification. Elephant seal milk is approximately 50% fat, not 3.5% like cow's milk, not 4% like human milk, 50. This milk is pale yellow, nearly opaque with fat, and has a consistency closer to thick cream than to anything pourable. If we were to put it in a container, it would not flow out clean at all. We could say it would be thick like very cold cream, and when it did move, it would do so reluctantly, lazily, in thick ribbons, leaving a visible coat on every surface it touched. The reason for this extreme composition is time pressure. A female elephant seal nurses her pup for approximately 28 days before returning to the sea. The pup cannot follow her into the ocean. It must build its entire blubber reserve, everything it will live off for months until it learns to feed itself, inside that one nursing window. The milk is not calibrated for slow development over years. It is a rapid energy transfer compressed in 4 weeks, delivering a fat load that any other mammal's digestive system would struggle to process. Dolphin milk sits at roughly 15 to 30% fat, depending on the species and the stage of lactation. It contains almost no lactose, the sugar that is the primary carbohydrate in cow milk and human milk. Marine mammal milk generally makes this trade: less sugar, more fat. Fat is a denser energy source per gram. A dolphin calf nursing for 18 months receives milk with more than 10 times the fat of human milk and a fraction of the sugar. The proportions reflect what the calf's body needs and what the ocean environment demands. The milk described in every carton you have ever seen and the milk a humpback whale delivers to a calf in open water are both, technically, milk. They share the same basic architecture: fat suspended in water, protein clusters floating in the liquid, sugars dissolved alongside them, minerals present in both. What they do not share is the proportion of any of those components. The range across all mammals is wider than any single image of milk can suggest. A single word covers a range of compositions that spans from the thin and sweet to the thick and almost buttery, each calibrated by millions of years of selection pressure to exactly the needs of the animal that produces it and the animal that receives it. Before continue, I want to tell you one thing. Subscribing to Sleepy Alchemy channel and leaving a like helps others find this kind of content. And let's continue. What human milk carries beyond nutrition is where the science becomes precise in ways that took much of the 20th century to understand. Human milk contains more than 200 distinct, complex sugar structures. That number is not unusual for a rich biological fluid. What is unusual is what these sugars are for. A human infant cannot digest them. They pass through the infant's own digestive system completely intact, unabsorbed, reaching the large intestine without being broken down at any earlier point. They are not in the milk to nourish the infant. They are there to feed specific bacteria. The bacteria in question, a species called Bifidobacterium infantis, is found in high concentrations in the gut of breastfed infants. It carries a very specific set of enzymes that can dismantle exactly these complex sugar structures. Other bacteria do not carry these enzymes and cannot access this food source. The milk in this arrangement is not simply feeding the baby. It is creating a selective environment inside the baby's gut, providing a food source available only to the bacterial species that should be dominant there, and unavailable to the bacterial species that should not be. The more than 200 complex sugars are a dietary instruction written into the milk that says in chemical terms, "These bacteria here and not others." The instruction is followed every feed in every healthy infant gut without announcement. No commercially available infant formula has yet replicated this selectivity. The enzyme substrate specificity involved is precise enough that approximating it requires knowing which sugars are present, at what concentrations, in what combinations, and at what stage of lactation. This was not understood until the late 20th century. For most of human history, and well into the era of modern nutrition science, these sugars were assumed to be inert, present but not important. It turned out they were among the more precisely constructed components of the liquid. The infant is not the only intended recipient of what is in the milk. The bacteria waiting in the gut are also recipients, and the milk was written for them too. It arrives at the large intestine, carrying food for residents it has never met, for a population it has been preparing for since before any specific individual was born. A property of milk that only became measurable recently sits alongside all of this. Milk produced at different times of day contains different levels of melatonin, the chemical signal the body uses to regulate the sleep-wake cycle. Melatonin in the mother's bloodstream peaks at night. Milk produced at night contains more of it than milk produced during the day. The difference is measurable in samples collected at different hours. Whether this melatonin, delivered through the milk at night, actually influences the infant's developing sleep patterns is still an open question. The mechanism is plausible, and the timing correlation is real. The full picture is not yet established. But the milk is, at minimum, chemically different depending on when it was made, and that difference follows the mother's own circadian rhythm. The evening feed and the morning feed are not the same liquid. A mother's milk delivers something different in the morning and something different in the evening, even on the same day. It is like coffee. When you drink it during the day, it gives you energy, but when you drink it in the evening, it can chase away your sleep. Yet both are the same coffee. It is just that the effect it has on you differs depending on the time of day you drink it. But coffee does the same thing, both during the day and in the evening. A person drinks coffee in the morning to get energy, but avoids it in the evening. This analogy may be a little confusing, but I tried to explain my thought to you in the simplest way that came to mind. As for the question of whether a mother's milk is truly different at night versus in the morning, this is still not fully known from a scientific standpoint. We cannot say anything definitive about it. The first milk a newborn receives is not the same liquid it will receive a week later. In the first 2 to 5 days after birth, the mammary gland produces a different secretion. It is thicker, more golden in color, and present in much smaller volumes than the milk that follows it. A mother produces perhaps a few teaspoons of this at each feed in the first day, against the many ounces of mature milk she will produce at peak lactation. It is called colostrum, and it is the reason the word milk doesn't quite cover what this gland can produce. The golden color comes not from beta-carotene concentrated in fat, but from the sheer density of immune proteins the fluid carries, yellow-tinted, heavy for its volume, unlike the white liquid that will replace it within days. Set against a white background, it carries a warmth in its color that mature milk does not, a visual record of what it is prioritizing before anything else. Mature milk is a nutritional fluid that also carries immune protection. Colostrum is an immune fluid that also carries nutrition. The ratio is reversed. Everything in the composition points towards the same priority, getting the newborn's defenses established before the gut is fully sealed and before the immune system has built its own library of responses. The protein concentration in colostrum is 2 to 3 times higher than in mature milk. Most of that additional protein is not structural or enzymatic. It is protective. The most abundant protective protein in human colostrum is a specific type of antibody, a molecule shaped roughly like a capital letter Y, with 2 arms that reach out and grip a specific target. In the bloodstream, these antibodies circulate as single units. In milk, they travel as double units, 2 joined together, wrapped in an additional protein coat that protects them from the acid environment of the stomach. This coating matters. Most proteins that enter the stomach are broken down by the acid and enzymes present there, dismantled before they can do anything useful further along the gut. This antibody is not. The coat holds. It arrives intact in the small intestine, the stretch of gut where most absorption and most pathogen contact occurs, and it attaches to the intestinal wall there, sitting along the mucosal surface like a row of guards posted at every gate. When bacteria or viruses come in contact with that surface, these antibodies bind to them. A pathogen that has been bound cannot attach to the gut wall. A pathogen that cannot attach cannot colonize. The defense happens at the surface before any systemic threat begins. These antibodies carry the mother's specific immune history. Every pathogen she has encountered, every vaccine she has received, every microbial environment she has lived in. The antibody library she has built over her lifetime is partially transferred through the colostrum into the newborn. An infant who has not yet had time to build its own immune memory receives a pre-assembled, highly specific collection of defenses on the first day of life. The transfer is liquid. It takes minutes. It cannot be replicated by any synthetic formula currently available, because the library it carries is specific to the mother's history and to the specific microbial environment the infant is entering. It is the most personal possible version of the letter, written from a single life, addressed to a single body, unrepeatable in any other combination. A second defense protein in colostrum works through entirely different logic. Lactoferrin is a protein found in colostrum in particularly high concentrations, as high as 7 grams per liter in early colostrum, compared to 1 to 2 grams in mature milk. It does not bind to pathogens directly. It does not carry memory. Instead, it does something more indirect and, in its own way, more elegant. It grips iron. Many of the bacteria that cause infant infections require iron to grow. Without iron, their metabolism stalls. They cannot divide. They cannot establish a colony. Lactoferrin binds iron with an extremely high grip strength, tight enough to hold it even in the acidic environment of the stomach, where weaker iron-binding proteins would release it. By moving through the gut and sequestering the available iron, lactoferrin makes the environment hostile to these bacteria without ever touching them. Lactoferrin does not kill bacteria. Bacteria need iron to grow, so it takes away the iron they need. Just as living creatures we can see with our eyes cannot survive without food, those bacteria cannot grow or spread without iron either. It is like taking someone's food from right in front of them without them knowing. This reminded me of a memory. Because I had not eaten enough and was still hungry, I quietly took a piece of food from my sister's plate while she was distracted by her phone, and because her attention was elsewhere, she never noticed. The gut it passes through looks the same afterward. Nothing was destroyed. Something was simply removed before it could be used. Lactoferrin also does something after it has been partially digested. When the stomach enzyme that breaks down proteins cleaves lactoferrin partway through, the fragments that result carry a different kind of antimicrobial property. They can disrupt the outer surface of bacterial cells directly, causing them to leak. The intact protein starves bacteria of iron. Its own digestion products attack bacteria through a different mechanism. The molecule is doing 2 separate jobs at 2 separate stages of digestion, sequentially from the same starting material. A single protein that becomes a different weapon as the body processes it. Alongside lactoferrin is a third molecular tool. Lysozyme is an enzyme, a molecular tool shaped to do one specific job. The job is breaking apart the outer wall of certain bacteria. Bacterial cell walls are built from a tough, repeating mesh structure, the way a chain-link fence is built from interlocking loops. Lysozyme finds a specific link in that mesh and cuts it. When enough links are cut, the wall weakens. The bacteria, which depend on their wall to hold their internal pressure against the surrounding environment, rupture. Lysozyme was first identified by Alexander Fleming in 1922, 3 years before he discovered penicillin, when he noticed that nasal mucus could dissolve bacterial colonies on a plate in the laboratory. The enzyme responsible is the same one present in tears, in saliva, and in human milk. Human milk contains approximately 3,000 times the lysozyme concentration of cow's milk. This is one of the reasons that early infant formula built from processed cow's milk was understood even before the full science was known to be nutritionally incomplete. A comparison of fat and protein and sugar across the two liquids could seem close. A comparison of lysozyme concentration was not close at all, and the difference was present from the first day. None of these proteins works in isolation. The combination of lactoferrin removing iron, lysozyme breaking bacterial walls, the antibodies binding to pathogen surfaces, the complex sugars feeding protective bacteria, and the slightly acidic chemical environment creates a gut milieu, a chemical atmosphere that is strongly oriented toward the health of the specific bacterial community the infant needs and against the pathogens that compete with it. No single component does this alone. The effect is the result of all of them operating simultaneously in the same liquid at the same time, each through a different mechanism, each aimed at a different target. The whole is considerably more than the sum of its parts, not because of any mysterious synergy, but simply because the threats are diverse and the defenses cover different ground. Something in the milk was not recognized until researchers looked at very high magnification. Fresh milk, particularly colostrum, contains living white blood cells, cells from the mother's own immune system present in the liquid, capable of identifying and responding to threats. In early colostrum, white blood cells can make up as much as 70% of the total cellular content of the milk. These cells can cross into the infant's gut tissue and function there. They carry with them the mother's immune memory, a more direct transfer than even the antibodies, because a living cell carries the full complexity of what it has encountered, the full library of responses it has been trained to make, the full biochemical toolkit of an experienced immune actor. These cells are not passive cargo. In the infant gut environment, research has shown that some of them retain the ability to migrate into the infant's tissues and take up residence there, at least in the short term. The mechanism by which they survive the transit through the infant's acidic stomach is not fully understood. What is clear is that they arrive in the gut in functional condition, and that their presence correlates with outcomes the antibodies alone cannot fully account for. The science on this is still developing, and some specific details of how the cells behave after crossing remain areas of active research. What is established is that the cellular transfer is real, that the cells are functional on arrival, and that their contribution to early immune function appears to extend beyond passive protection. Transferring a living cell is not like sending a message. It is closer to sending the messenger intact with all of their knowledge. The colostrum that looks from the outside like a few teaspoons of golden liquid is carrying at the cellular level the equivalent of a borrowed immune system, experienced, specific, and already trained for the environment the infant is about to enter. The letter, in this case, is not only written, it is delivered in person by someone who knows the territory. The fat inside a glass of milk is not one substance. Fat in milk is a mixture of hundreds of distinct fat molecules, each one built from the same basic components, but with chains of different lengths, different degrees of saturation, different points of melting. Because it is a mixture rather than a single pure compound, it does not have a single melting point. It has a melting range. At refrigeration temperature, some of the fat molecules inside each sphere have already solidified into tiny crystals, rigid, ordered structures floating in a pool of liquid fat within the same sphere. The sphere is simultaneously solid and liquid inside in proportions that shift with temperature. As the milk warms toward room temperature, the solid fraction melts into the liquid fraction. At body temperature, virtually all of it is liquid. This range, the transition from partly solid to fully liquid across a span of roughly 30 degrees, is not peculiar to milk fat. It is a property of all natural fats that are mixtures rather than pure compounds. Butter, lard, coconut oil all melt across a range. Refined industrial fats, engineered to be a single compound, melt at a single sharp temperature and have a hard, waxy mouthfeel that natural fats don't. If we take some butter and place it on a fresh piece of bread just out of the oven, it melts. We all know this. But why does it melt slowly rather than all at once? Because butter contains hundreds of different molecules, and each molecule has its own melting point. One melts at 10 degrees, another at 20, and another at 30. Each one has its own requirements. And because these molecules melt one after another in sequence, the butter softens gradually. The tongue registers the sum of all of this as smoothness, a single sensation produced by a process distributed across hundreds of distinct molecular thresholds happening in sequence. The chemistry of milk fat has one more unusual quality worth examining. Most of the fat we eat comes from sources dominated by long-chain fat molecules, chains of 16 to 18 carbon atoms. Olive oil, the fat in fish, the fat in most cuts of meat, all primarily long-chain. Milk fat is distinctive because it contains a meaningful fraction of short-chain fat molecules, chains of 4 to 12 carbons, at concentrations not found in most other natural foods. The shortest of these, a 4-carbon chain, is found almost exclusively in the milk of ruminant animals, cows, sheep, goats. It makes up approximately 3 to 4% of the total fat molecules in cow's milk. That may seem small, but this particular molecule is volatile. It evaporates at low temperatures, and the human nose can detect it at concentrations of parts per billion, a few molecules per billion molecules of air. Even cold, trace amounts of fresh butter evaporate from a fresh butter surface into the surrounding air continuously. The olfactory receptor that responds to this molecule is calibrated over evolutionary time to recognize it as something worth approaching. It is one of the primary contributors to the characteristic richness of warm butter and the familiar sharpness of aged cheddar. At concentrations in fresh butter, it smells appealing, warm, round, complex, the kind of smell that registers as nourishing before any conscious analysis occurs. At higher concentrations in poorly handled milk, it is the source of that sharp, almost sour note that announces something has gone wrong. The same molecule, the difference is quantity, a fragrance and a warning separated only by how much of it is present. Short-chain fat molecules also take a different route through the body after digestion. Long-chain fat molecules, after they are absorbed through the gut wall, are packaged into large transport particles, the body's way of moving fat through the lymphatic system before it reaches the bloodstream. Think of these particles as delivery trucks that need to be assembled at the loading dock before the journey can begin. The fat is loaded onto them at the gut wall. The truck is built around the cargo, and it travels through the lymphatic vessels before eventually entering the blood. This routing takes time and requires the assembly of those transport particles, which is metabolically involved. Short and medium-chain fat molecules, roughly 8 to 12 carbons, do not need this packaging. They cross the gut wall through a different path, entering the portal vein, the blood vessel that carries nutrients from the gut directly to the liver, without being assembled into transport particles first. They arrive at the liver faster. The liver processes them through a pathway oriented more toward burning them as fuel than toward storing them. Same liquid, two routing systems operating simultaneously, separating the fat into two metabolic streams based on nothing more than the length of the carbon chain. The body reads the molecular length and makes a routing decision from it, automatically, every time. It is a sorting system with no moving parts. The structure of the molecule is the only signal, and the signal is read correctly every time. The carbohydrate side of the liquid follows different logic entirely. The primary sugar in milk, present in cow's milk at approximately 47 grams per liter, and in human milk at approximately 70 grams per liter, is lactose. It is built from two simpler sugars joined together, glucose and galactose, linked at a single bond. At room temperature, lactose dissolves freely in the liquid, contributing a faint sweetness that is substantially milder than table sugar. Lactose is approximately 1/5 as sweet as sucrose by weight, which is why a glass of milk does not taste particularly sweet, despite containing a meaningful sugar content. The sweetness is present but restrained, the way a color can be present in a painting without dominating it. To use lactose as a fuel source, the body must first cut it apart into its two components. This cutting is done by an enzyme, a molecular scissor, produced by cells lining the small intestine. In most mammals, production of this enzyme decreases after the animal is weaned. The logic, from an evolutionary standpoint, is straightforward. An adult mammal in a world without dairy farming has no access to milk, and maintaining the machinery to process it is a metabolic cost with no benefit. The gene that controls production simply dials the enzyme down as the animal matures. In humans, this reduction happens in most of the world's population. Lactose intolerance in adults is the ancestral human state, not the exception. The genetic variant that keeps this enzyme active into adulthood, lactase persistence, is found in high concentrations in populations with a long history of cattle herding and dairy consumption. In northern European populations, the frequency of this variant reaches 90% or higher. In East Asian populations, it is below 5%. The mutation responsible in most European populations is a single letter change in the genetic code, a C replaced by a T in the regulatory region of the gene that controls enzyme production at a specific position. This single letter change keeps the gene switched on in adult tissue. It was essentially absent in European populations before the adoption of cattle herding, based on DNA recovered from ancient human remains. As herding became established, the mutation spread because individuals who carried it could digest milk as adults without symptoms, gaining a reliable caloric source that non-carriers could not reliably access. The genetic change did not cause herding. Herding created the conditions in which the genetic change carried an advantage, and the advantage spread the change through the population over thousands of years. Even though humans have changed many things, they did not change milk. On the contrary, milk changed humans. A single letter in a single gene changed. A T was written in place of a C. People who carried this change could still digest milk even as adults, and this gave them an advantage. Over thousands of years, this advantage spread, starting from a single letter and spreading across an entire continent. Lactose that reaches the large intestine undigested does not simply pass through. Even individuals with full enzyme activity do not absorb every gram of lactose that enters the gut. At large quantities, some passes through to the large intestine. There, bacteria that carry their own molecular scissors ferment the arriving lactose, producing short-chain fatty acids as byproducts. Those short-chain fatty acids are actually useful. They are a fuel source for the cells lining the large intestine, and they contribute to the mildly acidic environment of the colon that keeps the resident bacterial community healthy. The gas produced during this fermentation is the source of the discomfort associated with lactose intolerance. The mechanism is the same in all people. The difference between a comfortable experience and an uncomfortable one is a matter of how much lactose reaches the large intestine and what bacterial species are present to ferment it. It is a threshold difference, not a categorical one. The mineral content does something less visible than either. Calcium in milk, approximately 1.2 grams per liter, is not uniformly dissolved in the liquid. About 2/3 of it is bound inside the protein clusters, held there by bridges that lock the cluster's structure together, the way rivets hold metal plates, invisible from the outside but load-bearing throughout. The remaining third is floating free in the liquid phase. These two pools are not static. They exchange continuously. calcium moving from bound to free and back again, depending on the temperature and acidity of the surrounding liquid. In cold milk, more calcium sits in the bound form. As the milk warms, a fraction shifts toward the free form. No visible change marks this. The white does not shift, but the calcium is redistributing quietly at the scale of individual atoms. Every moment the milk exists. The exchange never pauses. It has no threshold at which it stops and rests. It is the kind of motion that belongs to matter simply being matter. No trigger, no signal, no reason other than that the temperature of the room permits it and the chemistry of the liquid requires it. It simply continues evenly beneath a surface that gives no record of it. Milk is not sterile when it leaves the mammary gland. Fresh milk drawn cleanly from a healthy animal contains a bacterial community, not a contamination, not a failure of hygiene, a resident population of microorganisms associated with the mammary gland tissue itself, present in the liquid before it contacts any surface in the external world. The bacteria most commonly found include species that are also found in healthy skin communities, the kinds of organisms that live quietly in the warm, moist environments of the body without causing any trouble. They are there before the milk is milk in any external sense. They are part of what the milk is. This was not widely appreciated until researchers began sequencing the complete genetic content of fresh milk samples, looking not for specific known bacteria, but for all bacterial DNA present, regardless of species. What appeared was not contamination. It was a consistent, reproducible pattern of microbial presence that matched the mammary gland's own microbial community, and that could not be accounted for by contact with external surfaces. The pattern appeared across samples from different animals, different farms, different seasons. It was the gland's own population, present with the same consistency as any other component of the fluid, as reliably present as the protein, as consistently distributed as the fat. The explanation researchers have proposed, still debated, still being tested, is that bacteria from the mother's own gut can travel through the body, carried inside certain immune cells, and be deposited into the mammary gland and secreted into the milk. The gut and the mammary gland communicating through a cellular messenger service with immune cells as the courier. Whether this pathway is real and physiologically significant in humans is an active area of research with genuinely uncertain current conclusions. What has been established is that the bacterial community in fresh milk is consistent and not random, and that it resembles the community found in the breastfed infant gut more closely than chance alone would suggest. The milk, if this hypothesis holds, is not just delivering chemistry, it is delivering residence, a starter population, seeded into a new environment before it has developed its own. Once outside the body, the fate of that community is governed entirely by temperature. At refrigerator temperature, 4 degrees Celsius, the bacteria in fresh or pasteurized milk are not inactive. They are metabolizing, but slowly. Most of the species present prefer temperatures between 25 and 37 degrees, where they would double in population every 20 to 30 minutes. At 4 degrees, their doubling time extends to many hours. The chemistry they produce, acids, enzymes, flavor compounds, accumulates far more slowly. This is all refrigeration is doing, not stopping the process, slowing it to the point where the product remains palatable within a practical window. The refrigerator does not stop the processes happening inside milk. Cold is not the same thing as pressing a stop button and shutting down or halting some kind of mechanism. Here, the cold simply slows down the processes taking place inside the milk. It is as if someone has tied a thick rope around them and is trying with all their strength to prevent the processes moving forward at speed. As if it is trying to turn red lights on along the road so they fall behind. No matter how hard the cold tries, these processes will continue, slowly, but they will continue. One enzyme naturally present in fresh milk has a thermal behavior specific enough that the dairy industry uses it as a built-in indicator. Alkaline phosphatase is a protein found in the fat sphere membranes and in the liquid phase of fresh milk. It is inactivated by standard pasteurization temperatures. 72 degrees Celsius for 15 seconds destroys its activity reliably. This relationship between the enzyme and the heat treatment is so consistent and so well documented that testing for the enzyme's activity in a supposedly pasteurized batch is a standard quality control step. Active alkaline phosphatase means the milk was not pasteurized to the correct temperature, or that raw milk has been added afterward. The enzyme is not harmful. It is simply a thermometer that cannot be faked, a chemical record of whether the correct temperature was reached, readable from the liquid itself long after the heat treatment is over. No document, no label, no verbal account is required. The milk carries its own audit, available to any laboratory that knows how to read it. The record is in the molecule. The molecule either functions or it doesn't. There is no ambiguity. Something else in the milk was not visible until electron microscopy advanced far enough to find it. Milk contains extremely small membrane-wrapped particles, so small that thousands of them lined up end to end would barely cross a millimeter. These particles are shed by the cells that produce the milk, and they carry a cargo: proteins, lipid molecules, and small fragments of genetic material. Not the full genetic code, fragments, specifically the kind called microRNA, which are short sequences that can influence which genes in a recipient cell are active without changing the DNA itself. Think of them as annotations written in the margins of a book, not rewriting the text, but flagging certain passages, telling the reader to pay closer attention here, to skip past there. The book remains the same. The reading of it changes. These particles survive the journey through the milk. Whether they survive digestion and reach infant cells intact is still being determined. Laboratory studies have shown that similar particles from cow's milk can be taken up by human gut cells. Whether this happens at physiologically meaningful levels in a living infant, and whether the microRNA they carry actually influences gene expression in the baby's developing systems, is genuinely unresolved. The particles are real. The cargo is real. The physiological significance in a living organism is the open question. This is not a gap in the science. It is the frontier of it. the place where the tools are new enough to see the question clearly for the first time, but not yet powerful enough to answer it completely. The fact that the question can now be asked precisely is itself a recent achievement. Within a single nursing session, the milk itself changes. The milk at the beginning of a nursing session is not the same as the milk at the end. Early in the feed, what flows is lower in fat, around 1 to 2%, and higher in water, lactose, and proteins. By the end of the session, as the gland contracts more completely and expresses milk from closer to the secretory cells, the fat content rises to 5 to 8%. The total fat delivered in a single feed depends not just on how much milk is consumed, but how completely the feed is completed. The reason for this gradient is mechanical and physical, not hormonal. Fat spheres, being less dense than the surrounding liquid, collect preferentially near the walls of the gland's internal ducts during the resting period between feeds, floating gently toward the surfaces, clustering there in the quiet interval, waiting. When flow begins, the less fatty liquid from the larger collection ducts comes first. As the smaller ducts and secretory cells are reached and expressed, the fat-rich liquid comes last. The gradient is a consequence of density and duct geometry, not a designed feature. It was not engineered into the system. It emerged from the physics of fat in a liquid in a tube, operating continuously and passively between every feed. A physical phenomenon that happened to become nutritionally meaningful. The end of a feed richer in energy density than the beginning, not by design, but by the behavior of fat in a gravitational field. In milk, homogenized in a factory, where the fat spheres have been broken into fragments too small to cream and too evenly distributed to form any gradient, this distinction does not exist. The foremilk and hindmilk are features of the gland. Outside the gland, in any processed form, they are gone. After the milk is sealed in a container, a different kind of evolution continues. Certain enzymes in milk, proteins that act as chemical catalysts, continue their work after processing and packaging. One group of enzymes naturally present in milk can cleave fat molecules, releasing the short-chain fatty acids that contribute to flavor and, at elevated concentrations, to off flavors. These enzymes are normally kept separated from the interior of the fat sphere by the intact membrane around each sphere. If that membrane is disrupted by rough handling during transport, by freezing and thawing, by aggressive pumping in processing equipment, the enzymes gain access to the fat they were previously blocked from reaching. The short-chain fatty acids that result are released into the liquid. At trace concentrations, they are part of the complex flavor of good butter and good cream, a background note of richness that experienced tasters recognize, a warmth in the flavor that sits below the threshold of identification but contributes to the overall impression. At higher concentrations, they produce a sharp, rancid note that overtakes everything else. The membrane that originally wrapped each fat sphere, 3 molecules thick, is what stands between those two outcomes. 3 molecules is not a measurement that registers as meaningful in ordinary life. It is smaller than anything the hand can feel or the eye can see, and yet it is the entire distance between a liquid that tastes clean and cold and right and one that does not. The difference between milk that tastes right and milk that doesn't is sometimes that thin. 3 molecules of separation between the appealing and the alarming. The whiteness of milk is a physics event, and the physics can be described with precision. White light is a mixture. Every color visible to the human eye, from violet at the short end to red at the long end, travels as a wave, and each color has a characteristic wavelength. Violet light waves are approximately 380 nanometers long. Red light waves are approximately 700 nanometers. All the other colors fit between these two. A nanometer is 1/billionth of a meter, a unit so small that 80,000 of them span a single human hair. The fat spheres in milk range from approximately 100 nanometers to 10,000 nanometers in diameter. They live, in other words, directly in the size range where light waves and particles are comparable to each other. And that is precisely the relationship that determines what happens when they meet. When a light wave encounters a particle whose size is close to its wavelength, something interesting happens. The light cannot simply sweep around the particle as if it were not there, and it cannot bounce off a flat surface cleanly the way a billiard ball bounces off a cushion. Instead, the light interacts with the charged particles inside the material, and they re-radiate the energy outward in all directions, the way a stone dropped in water sends ripples outward in every direction simultaneously, with no preference for any particular angle. This is scattering. The physics of this interaction, how the size of the particle relative to the wavelength determines the pattern of scattering, was fully worked out by the German physicist Gustav Mie in 1908. A fat sphere in whole milk is close to the wavelength range of visible light, and it scatters all visible wavelengths with approximately equal efficiency. Do this simultaneously across 10 billion spheres in a glass, and the result is a white that has no preference for any color, the complete, even white that makes milk what it looks like. The atmosphere scatters light too, but by a different mechanism. Gas molecules, oxygen, nitrogen, are far smaller than fat spheres. At that smaller scale, the physics shifts. Shorter wavelengths scatter much more strongly than longer wavelengths. Blue light scatters approximately 10 times more intensely than red light from particles at that size. The result is that the entire sky above fills with scattered blue light during the day. Sunsets are red and orange because when the sun is near the horizon, its light must travel through a much thicker slice of atmosphere to reach the eye. The blue has scattered away in all other directions long before it arrives. The red and orange, which scatter less, survive the long journey. These are not two separate phenomena. They are the same physics operating in two different size regimes, producing two different appearances from the same source of light. The milk in the glass and the sky above the building containing it are both displaying the same underlying relationship between particle size and wavelength, separated only by scale. Skim milk sits between these two regimes. Its protein clusters are smaller than the fat spheres of whole milk, and they scatter blue light preferentially, not as strongly as atmospheric gas molecules, but with a measurable preference. The faint blue cast of skim milk in a clear glass in natural light is the same physics as the sky, scaled down to a few centimeters of liquid. Remove the fat from whole milk, and what is left is a thin slice of what the atmosphere does, contained in a glass. The physics that produces a summer sky and the physics that produces the color difference between whole and skim milk are written from the same equation. The fat spheres have another optical property that changes with temperature in a way most people would not expect. The refractive index of a material is a measure of how much it slows light down relative to the speed of light in a vacuum, a number that describes how much the material bends light as it crosses a boundary. Water has a refractive index of approximately 1.33. The fat inside a milk fat sphere has a refractive index of approximately 1.46. That difference, the gap between the two values at the boundary between fat and water, is what causes light to scatter rather than pass through. If the fat had the same refractive index as the surrounding water, it would be invisible. The light would cross the fat-water boundary without noticing the boundary was there. The fat sphere would still be present physically. It would still occupy space, still drift in its Brownian walk, but it would contribute nothing to the whiteness. The milk would be as clear as water. Every drop of whiteness in every glass of milk that has ever been poured is entirely dependent on a gap of 0.13 between two numbers, less than 0.2 of a unit. But without it, the color is gone. As fat warms, it expands slightly, and its refractive index decreases. The gap between the fat's refractive index and the water's narrows. At around 40 to 50 degrees Celsius, the gap has narrowed enough that scattering efficiency drops noticeably. Warm cream becomes visibly more translucent than cold cream, not fully transparent, but measurably different, the whiteness thinning toward something closer to an off-white haze. The whiteness of milk is not fixed, it is temperature-dependent, traceable directly to how the refractive index of fat responds to warmth. The opacity of warm cream is measuring the same temperature as a thermometer would by different means, light entering the liquid and failing to scatter cleanly as the refractive gap narrows. The color in a glass of milk changes as the glass warms, imperceptibly slowly, never reaching transparency, but moving toward it. The calcium that holds the protein clusters together is worth examining at the atomic scale. Calcium is element 20 on the periodic table. Its nucleus contains 20 protons. Its outermost 2 electrons are held loosely, further from the nucleus than the 18 below them, weakly bound, the way a satellite held at the edge of a planet's gravitational field is held less firmly than one in a close orbit. In a water solution, these 2 electrons are gone. Calcium exists as a doubly charged ion. It has given away 2 electrons and carries a double positive charge as a result. That double positive charge is the reason calcium does everything it does in milk. It binds strongly to negatively charged groups on the surface of proteins, bridging between them the way a staple bridges 2 sheets of paper, holding them in contact under any ordinary force. In the protein clusters of milk, multiple calcium ions bridge multiple protein chains simultaneously, holding the entire cluster together in a stable, porous assembly. The cluster is not a solid block. It is an open, sponge-like structure, more empty space than material, held in its shape by these calcium bridges at intervals throughout. Each bridge is a single calcium ion holding two protein chains in proximity, a relationship maintained not by physical contact alone, but by the mutual satisfaction of opposite charges meeting across a small gap. Remove the calcium, strip it out of the liquid with a substance that binds it more strongly than the proteins do, and the clusters fall apart. The proteins remain intact. The liquid remains white because the proteins still scatter light, but the structure that gives milk its behavior during fermentation and cheese making is gone. The bridges that held everything in place have been taken, and what remains disperses. In bone, calcium combines with phosphate to form the mineral that gives bone its compressive strength, the same material that forms tooth enamel. This mineral is deposited in a very specific orientation along long protein fibers, the fibers oriented along the axis of mechanical load, the mineral crystals arranged around them perpendicular to that axis. The result is a composite, flexible fiber running in one direction, hard mineral surrounding it, the two working against different types of force at the same time. Neither component alone could resist both bending and compression. Together, they can do what neither could separately. The calcium in a glass of milk, if absorbed through the gut wall and deposited in bone, will participate in maintaining this architecture, arriving as a dissolved ion, depositing as part of a crystal lattice, contributing to a structure that the body will load mechanically for decades. Bone struck lightly with a knuckle returns a faint, dull sound, dense, unresonant, nothing like the ring of metal or the hollow tap of wood. That particular quality of density is partly what calcium built, atom by atom, deposited from liquids drunk years or decades before the knock. Below the fat sphere scale, the atoms themselves are in motion. The lactose molecules dissolved in the liquid contain 45 atoms each, a small, complex shape that has been described earlier as a pair of interlocked rings. At room temperature, these molecules are moving through the liquid, being redirected continuously by collisions with the water molecules surrounding them. Each water molecule is moving at several hundred meters per second on average, and each collision changes the lactose molecule's direction slightly. The cumulative result is a slow drift, a net movement that takes days to carry a single lactose molecule from the bottom of a glass to the top by thermal motion alone. This is not a failure of physics. It is the physics of particles in a medium. The drift is called diffusion, and it operates at this slow pace precisely because the liquid has density and resistance. Diffusion in air is much faster. In a dense liquid, the same molecular process is slowed by the friction of the medium, like trying to walk through a crowd rather than an open field. The lactose molecule drifts anyway. It has no choice. The thermal energy that drives it never diminishes at room temperature. It simply arrives at its destination more slowly than it would in a thinner medium. The fat spheres are undergoing the same drift, their Brownian walk in the first chapter, but at a different scale. The fat sphere is vastly larger than the lactose molecule, and therefore moves more slowly on average. The lactose molecule, being smaller, moves faster. The mineral ions dissolved in the liquid, smaller than both, move faster still. The entire interior of a glass of milk at rest is a system of continuous motion at every size scale. Atoms, molecules, clusters, fat spheres, all in motion, all at different rates, none of it visible from the surface. If we pour milk into a glass and look at it, we see nothing but white milk, right? But even though we cannot see it, inside the milk, atoms, molecules, fat globules, in a word, everything is in motion, all of it moving without pause. There is a quality that all of the properties examined across these chapters share, and it is worth naming before everything settles. None of them announce themselves. The saliva reading of the first chapter, the milk adjusting its composition within hours of feeding, leaves no visible trace. The color of the liquid does not change, the temperature is the same, the volume is the same. The immune proteins added to the next feed are present at concentrations too small for any sensory organ to detect. The event is chemical and precise and invisible, and it happens in the ordinary course of something that looks like the simplest possible exchange between two bodies. This is the recurring quality of milk at every scale examined so far. Whatever it is doing, it does not show from the outside. The golden colostrum that carries a lifetime of immune memory looks like a slightly yellowed fluid. The 200 complex sugars aimed precisely at a single bacterial species look like dissolved sweetener. The membrane 3 molecules thick around each fat sphere, the barrier separating good flavor from rancid, the original coat from the substitute, is invisible from any external observation. The letter does not advertise its contents. It has never needed to. The contents arrive regardless. The casein protein clusters, the protein assemblies that give milk much of its structure, are held together by calcium bridges, as described in the previous chapter. What was not previously mentioned is the nature of those bridges at the level of individual atoms. Calcium ions, carrying their double positive charge, bind to specific sites on the protein chains. Those sites are amino acids carrying a phosphate group attached to them. Phosphate is a combination of one phosphorus atom surrounded by 4 oxygen atoms, the oxygens carrying negative charges that attract the positively charged calcium ion, the way opposite poles of 2 magnets draw together. The bridge is 2 protein chains by being attracted simultaneously to a negatively charged phosphate on one chain and a negatively charged phosphate on another. The bridge is not a covalent bond, not the kind of bond where atoms share electrons. It is an electrostatic attraction, the same force that makes a balloon stick to a wall after being rubbed. Strong enough to hold the cluster together under ordinary conditions, not so strong that it cannot be broken by a change in the surrounding chemical environment. This is the reason milk's behavior changes so dramatically when it acidifies. As the liquid becomes more acidic, more positively charged hydrogen ions accumulating in the solution. Those hydrogen ions compete with the calcium ions for the same negatively charged sites on the protein chains. The calcium is displaced. The bridges break. The protein clusters lose their structural glue and begin to aggregate with each other instead of remaining dispersed. The liquid, which was pourable and white, becomes a gel. The process is gradual and then sudden. The buffering chemistry of the liquid holding the system stable right up until the point where the calcium bridge count falls below the threshold needed to maintain dispersion, and then the clusters contact each other all at once. Just because a process or event cannot be seen with the eye does not mean nothing is happening there. Sometimes we are not aware of it, but when the result occurs, we are sometimes surprised. How did this happen all of a sudden? In reality, it does not happen all of a sudden. It happens slowly. Milk is exactly the same. At first, it looks fresh, and then suddenly we see it has already curdled. Someone unaware of these processes would most likely say it happened all at once. In reality, these processes were already underway. We simply see the result. It is like achieving success in life. Even if no one sees it, you work alone day and night for years, and finally you receive the result of all those years of struggle in a single night. But those watching from the outside, unaware of all the effort and sacrifices you made, only talk about the result that appeared all of a sudden. This is yogurt. This is the first stage of cheese making. It is the destabilization of an electrostatic architecture, a structure that held because of the charge on individual atoms and fell when those charges were competed away by something smaller. The phosphorus chemistry that runs alongside calcium is doing something equally quiet. Phosphorus in milk exists in 3 different chemical forms simultaneously. It is present as a free ion dissolved in the liquid phase. It is present as a structural component of the fat sphere membranes, part of the molecules that form the organized skin around each fat sphere, their heads pointing outward toward the water, their tails pointing inward toward the fat. And it is present as the phosphate groups attached to the protein chains, the sites where calcium bridges. These 3 forms of phosphorus are chemically distinct. They do not exchange with each other under normal conditions. They are 3 separate jobs assigned to the same element, operating in 3 separate locations in the same liquid, none of them interfering with the others. This is one of the less visible examples of molecular economy in milk. The same atom, phosphorus, participates in the membrane architecture, the protein cluster architecture, and the free mineral pool, all at once in the same glass. It is doing 3 things simultaneously without any of the 3 being aware of the others, because atoms are not aware of anything. They are simply in the positions that the available energy permits. The arrangement is not designed. It is the accumulated result of what worked being retained and what didn't being lost over an enormous stretch of time. At the smallest scale at which anything in the glass can be described, the picture changes again. The bond between 2 atoms, any 2 atoms, anywhere in the liquid, is not a physical connector. It is not a wire or a hinge or a thread. It is a region of space where the probability of finding the electrons from both atoms is elevated. Electrons in quantum mechanics do not orbit in defined circles. They exist in probability distributions, regions where they are more likely to be found without any certainty about their exact location at any given moment. Think of the electron not as a small ball moving along a track, but as a smeared presence, a cloud of likelihood that is denser in some regions and thinner in others, never pinned down to a single point. A bond forms when the probability clouds of electrons from two atoms overlap in a way that lowers the total energy of the system. The overlap is stable because any movement away from it would require energy the system doesn't have at that temperature. Nothing in this description chooses. No atom decides to stay. The arrangement persists because the geometry permits it and the energy requires it. This is all that stability means at any scale. The calcium bridge between two protein chains is not a solid rod. It is a region of overlapping probability distributions, the calcium ion's electrons and the oxygen atom's electrons, finding a shared spatial arrangement that costs less energy than the separated alternative. The entire structure of the casein protein cluster, the bridges, the protein chains, the phosphate groups, the porous interior where smaller molecules move freely, is a system of these overlapping probability arrangements, each one stable because the alternatives cost more energy, collectively producing a structure that is robust enough to survive pasteurization, flexible enough to disassemble in acid, and complex enough to carry and protect the liquid that surrounds it. Stability, in this sense, is not rigidity. It is the lowest available energy state, held until something offers the system a lower one. Everything solid in the glass rests on that principle. Every structure persists because moving would cost something the system does not currently have. What remains genuinely open in this examination is worth naming before it closes. The platypus's monotreme lactation protein, the spiral-structured antimicrobial protein identified between 2010 and 2019, has a confirmed structure and a confirmed antimicrobial effect. The precise molecular mechanism by which it produces that effect is not yet established. The enteromammary pathway, the proposed route by which bacteria from the mother's gut travel via immune cells to the mammary gland, has evidence supporting it and evidence complicating it. The question of whether milk exosomes deliver functionally significant microRNA to infant cells in a living organism rather than in a laboratory dish does not yet have a settled answer. The influence of nighttime melatonin in milk on infant sleep development remains a correlation without a confirmed mechanism. These are not failures of the research. They are the questions the field is currently working through in real time with the tools available. Scientific uncertainty is not the same as scientific ignorance. What is uncertain is known to be uncertain. What is established precisely. The two sit alongside each other without contradiction, confident knowledge and admitted open questions, held together without pretending either is the other. That combination is what the subject deserves, and it is, for the kind of listening that this channel is built for, more interesting than false certainty would be, because an honest, open question has weight in a way a manufactured answer never does. If one of these details stays with you, it may be worth leaving in the comments. The quietest observations often turn out to be the most interesting ones. The surface of the glass that started all of this is still there. A white, cool, slightly heavy liquid. A surface that scatters light from 10 billion fat spheres simultaneously and returns it to the eye as an even, sourceless white. a foam that forms at the top when it is disturbed and holds for a minute before the protein walls of each bubble thin to the point of breaking. A weight in the hand that is slightly more than water and slightly less than the richness of cream. A smell that is neutral and faintly sweet, and if the milk has been kept cold and the fat sphere membranes are intact, entirely without off-note. It is cool against the palm. It is quiet when still. It asks for nothing. Everything examined across these chapters is hidden within that surface. The calcium bridges holding the protein clusters together, the fat spheres walking their unpredictable thermal paths through the liquid, the lactose molecules drifting toward equilibrium at a rate too slow to see, the phosphorus in 3 simultaneous jobs, the protein that sequesters iron from bacteria before they can reach it, the antibodies that survived the stomach and are sitting on a gut wall somewhere, waiting for a pathogen that may never arrive, the bacteria that were present before the liquid left the body, the particles carrying fragments of genetic instruction toward a destination that research has not yet fully mapped. All of it is there, none of it visible. The glass looks like a glass of milk. The letter does not show its contents from the outside. It never did. The envelope is simply white, cool even, sourceless in its glow, and everything written inside it waits at the scale that the eye cannot reach, patient in the way that matter is always patient, moving in the way that matter always moves, existing quietly in the only configuration available to it at that temperature, in that glass, in this particular moment of examination. There is no fanfare for what is happening inside. There is only the white surface, the quiet weight, and the scale at which all of this becomes visible, which is not the scale of the hand or the eye or the room, but the scale of the thing itself, smaller than any instrument most people have ever held, present in everything poured from a jug on an ordinary night. There is a further examination waiting of a material that shares some of this liquid's molecular strategies and developed them in a completely different environment, with a history of its own that runs alongside the history of milk without intersecting it. That examination does not begin tonight, but it begins. We have reached the end of the video. If you want to know more interesting facts while falling asleep, you can find more on the Sleepy Alchemy channel. Good night. See you in the next video.